Okay, hello everybody, and good to see so many of you here. For those of you who haven't joined me before, my name's Tom. It's A-levelbiologytutor.com. Today we're going to go over the nervous system. Should take about 45-50 minutes. We'll do a Socrates quiz about 2/3 of the way through. I hope you can all hear me. If you can't, try switching on your speakers a bit louder, and anytime you want to have a chat or want to ask a question, then it's in the Q&A box. You just go up to that at the top of the screen and type it in. So let's make a start. Let's go through this. So, nervous system, we've got two parts to it. We've got the central nervous system, which is the brain and just the spinal cord. And then we've got the peripheral nervous system that's coming away from that. Now, the peripheral nervous system is made of the sensory and the motor neuron. The sensory neurons carry information to the central nervous system, and the motor neurons carry it away to an effector, which is a muscle or a gland. Now, concepts of receptors. So receptors we have are things like rods and cones, the olfactory cells in the nose, the taste buds in the tongue, Pacinian corpuscles in the skin, we're going to talk a lot about, and sound receptors in your ear, and then proprioceptors in your muscles to check the length of your muscle fibers and show where your bones are. So we're going to look now at the structure of a neuron. So things you need to know. You definitely need to know the structure of a motor neuron. Now, the structure of a motor neuron is coming from a relay neuron. So you have a relay neuron connected by a synapse, and then you have dendrites, and the dendrites go past the cell body. Now the cell body contains the nucleus and the endoplasmic reticulum and the mitochondria. And then we have, going away from the cell body, you have the axon, and the axon has in it axoplasm, and around the cell around the outside of it is the axolemma, which is the cell membrane. Now, with motor neurons, you have Schwann cells, and these are cells that are wrapped around the axon. Remember, the axons of motor neurons can be very long. So you've got a motor neuron going from your spinal cord to the end of your finger to contract the muscles in the end of your finger. So that's one cell. The Schwann cells are there to increase the speed of communication by allowing saltatory conduction, which occurs between the nodes of Ranvier, so that the action potential can jump its way down the axon. Now, the Schwann cell contains myelin, which is a lipid, and it's an insulator, so it's electrically insulating the neuron. The motor neuron goes from the dendrites past the cell body, down the axon, and then to the axon terminals. And the axon terminals connect to a neuromuscular junction or to an effector such as a gland. So they carry the impulses from the central nervous system to the effectors, and this is what you need to know. You need to know there's a cell body, you need to know there's an axon that goes away from the cell body, and those end up in axon terminals. So the axon conducts the action potential away from the cell body. The axon terminals release the neurotransmitter. The neurotransmitter is a chemical messenger which will be picked up by a receptor, which is on the postsynaptic junction or on the other side of the neuromuscular junction. Then you've got dendrites, and dendrites carry the impulse towards the cell body. Cell body contains nucleus, mitochondria, ribosomes. And then you've got the myelin sheath, which is the fatty acid insulating layer. Okay, then you've got a dendron, Schwann cells, node of Ranvier. Node of Ranvier is the area along the axon where you don't have the myelination. And that part is where the axon is exposed and where an action potential can occur. So we're going from a sensory neuron. Now the sensory neuron is going from a dendrite past the cell body, and the cell body is going to be in the dorsal root ganglion, and then it becomes the axon. So you have dendrite, dendron, cell body axon going to axon terminals, and these axon terminals will junction with a relay neuron. So the sensory neuron brings the impulses from the sense organ into the central nervous system. So if these were the dendrites, this is where you'd have, say, a touch receptor, such as a Paxinian corpuscle, and we'd send the action potential along the dendron and then along the axon after it's gone past the cell body. Relay neuron, this is known as a connector or an interneuron in many syllabuses, and this connects a sensory neuron to a motor neuron if it's just a reflex arc, or to connect to other relay neurons. So these are connector neurons, and because of that, they don't have myelination around them because they're only transmitting it a very short distance. So if we look at a reflex arc, so we've got a receptor, and the receptor, if we take as an example, touch receptor, so we'd have a Paxinian corpuscle around the end of the dendrites. We send the action potential if we've achieved a higher than threshold level stimulus. Then we start an action potential, it travels along, it goes through the dorsal root ganglion and then into the connection, which is the synapse between the sensory neuron and the relay neuron. Now, when you get to a synapse, you're converting an electrical signal into a chemical signal, which is the release of the neurotransmitter, which diffuses across the synapse, which is the name of the gap. And then it gets transmitted to the motor neuron and then leaves through the ventral root and goes out to the effector, which is the muscle or the gland. Now, this section in the middle is often called the grey matter, and a very frequent exam question is, why is the grey matter grey? And the answer for that is that that's where all of the cell bodies are, and that's where there is no myelination because they're all relay neurons. So if you hear your parents say, use your grey matter as an aphorism about thinking about it, well, actually they're more correct than they think, because your brain is all grey matter because it's all relay neurons. So here's our reflex arc. Remember, reflex arc is protective and involuntary action. And the reflex arc consists of something coming from a sensory neuron, going through the relay neuron, and then out through the motor neuron through the ventral root. So a rapid involuntary response doesn't involve the brain, and move on through that, move on, skip on through that, skip on through that, skip on through that. Here we go. Right, stimulus, receptor, dorsal root ganglion, cell body of the sensory neuron, synapse, relay neuron, motor neuron, and the cell body is here, the cell body is here, and the cell body is here. Do remember, you do need to label the cell bodies, you do need to label the grey matter, you do need to label the dorsal root ganglion, you do need to label the ventral root, and ventral is front, dorsal is back. If you think of the dorsal fin on a shark, that's on the back of a shark. The dorsal side is the back and the ventral is the stomach side. Okay, so now let's look at the transmission and generation of an action potential and the transmission of a resting potential. So these experiments were done on squid axons. Now, they're not the axons of giant squid, they are large axons, because squid are carnivores. They need to have a very high speed of transmission of the action potentials, but they've got unmyelinated axons. So the way they achieve a high speed of transmission is they have a low resistance by having a very large diameter, which means that you can get electrodes into the axon, so into the middle of the axon or onto the side, so you can measure the electrical potential, the potential difference across the axon. So how do we create a resting potential? So with a resting potential, the way you do this is you have a axon. So we have cell membrane and cell membrane. Then inside the axon, we have axoplasm. The axoplasm is more negatively charged than the tissue fluid because of the presence of lots of negatively charged proteins and anions. And then we've got our sodium-potassium pump. Now, if you remember the kidney, you've got a sodium-potassium pump there. It's exactly the same thing. It pumps sodiums out and it pumps potassiums in. So it pumps 3 sodiums out. Now, because it's a pump, it's using energy. It's using energy in the form of ATP. So it pumps 3 sodiums out and 2 potassiums in. And in order to do that, it's hydrolyzing ATP into ADP. Now that means we get lots of sodiums outside and few sodiums inside. And you get lots of potassiums inside and you get few outside. Now the membrane is more permeable to potassium than it is to sodium. So more of the potassiums leak out again than sodiums leak in again, which means that overall we have more positive charges outside and more negative charges inside, which means that we then create a potential difference across the membrane. Now that potential difference is about minus 70 millivolts. And that's known as the resting potential of the neuron, which is the difference between the axon and the tissue fluid that's outside of it. Now, what happens next is that's perfectly fine until you raise the potential. So if you look at a Paxinian corpuscle, and this is a touch receptor. So Paxinian corpuscle, we've got layers of membrane, and when we deform the Paxinian corpuscle, you allow in some sodiums. So if you put pressure on the Paxinian corpuscle, and this is just a touch receptor, you're allowing in some sodiums. Now those sodiums are positively charged. So what's going to happen is that the resting potential is going to become less negative because you're allowing in more sodiums. Now, if you stay below the threshold level, then nothing will happen because the threshold level is at about minus 55 millivolts. And at minus 55 millivolts, that's the potential difference that needs to be reached before a voltage-gated sodium channel opens. Now, if you are below the threshold level, then that channel will not open. So these are known as sub-threshold level depolarizations. But if you touch something enough that you allow through enough sodiums, then you reach the threshold level, and at that point, the voltage-gated sodium channel opens and sodiums flow through facilitated diffusion down their gradient into the cell. Now that influx of sodiums leads to the inside becoming positive. Now at this point, remember, the potassium channels are closed. So during the action potential, which is this period, so we've reached the threshold level, we've opened the sodium channels, the voltage-gated sodium channels have opened, the sodium's come flooding in, the inside is now positive, and the potassium channels are closed. Now the sodium channels only open for about 1 millisecond, and then they snap closed. Once they've snapped closed, the sodiums stop entering. Now the influx of those sodiums will mean that the next bit along of the axon, that next bit along, remember, is still negatively charged because there are more sodiums on the outside. So you've now got a situation where, because the sodiums have come flooding in, which has caused the action potential, the sodiums have come flooding in here. Well, these sodiums then move towards where there are less sodiums. And these sodiums here move this way because it's more negatively charged. So you've got the positive charges move towards where there are more negative, slightly more negative charges and less sodiums, and these ones move this way. And this sets up what's called a local circuit. So if we think about what the potential difference is at this point here, that point, it's going to be below the threshold level. And as the sodiums move this way, because there's been an influx of sodiums because you've reached the threshold level because the voltage-gated sodium channel's open, so the sodiums have been flooding in, well, those sodiums will be attracted this way along the axon. And these ones here will be attracted more this way. So that will mean that the next part of the axon along the potential difference will rise, and it will rise over the threshold level. So the next voltage-gated sodium channels will open, and then more sodiums will come flooding in at that point, because the next part of the axon reached the threshold level. Now, because those have come flooding in, well, now the inside is positive at this point. These will be attracted this way towards where it's negative because it's still at a resting potential further along. These will be attracted this way because it's now negative. So this is how the wave of depolarization spreads down the axon, because the influx of sodiums causes the next part of the axon to be over the threshold level, which opens the next sodium channels, which allows in more sodiums, which opens the next ones, which allows in more sodiums, which opens the next one, and so on. So the wave of depolarization will spread down the axon. Okay, I hope that made vague sense. Now, what happens after you've had the influx of sodiums? So we got over the threshold level, we opened the voltage-gated sodium channels, the sodiums came flooding in down their potential gradient, and the reason why there's a gradient is because you actively pump them out. So we had the sodiums come in, which makes the inside more positive. Then, after the sodiums have come in, remember we've been over the threshold level, sodiums come flooding in, then the sodium channels close. And the next thing is that voltage-gated potassium channels open. So these voltage-gated potassium channels respond to the peak of the action potential here. So at that point they open, and they allow the potassiums out, and these travel through facilitated diffusion from the inside of the axon to the tissue fluid outside, and that repolarizes the membrane below the threshold level. Now, at this point here, when you've had an action potential, remember the voltage-gated sodium channel opened, and it opens only for 1 millisecond. And then it undergoes what's called a refractory period. So this is when it won't respond no matter what the potential difference is across the membrane. So from here to here, you're over the threshold level. So the voltage-gated sodium channel should open. But because it's in a refractory period, it's closed, and it remains closed until the end of the refractory period. And by the end of the refractory period, you're then below the threshold level. So that then the refractory period ends and the sodium channel is able to respond, but you're below the threshold level. Now, the efflux of potassiums, the potassiums leaving, going again from where there are lots to where there are few, that depolarizes the membrane. But now, when we're down here, we've got lots of sodiums inside and lots of potassiums outside. So at that point, you then need to restore the resting potential by pumping the sodiums that have come in out and pumping the potassiums that have gone out back in again, which is what happens here when the axon is hyperpolarized. Good. Let's go back to the PowerPoint. Okay, are we still there? We're all there, lots of you. It's good to see you all. Right, so let's go back to PowerPoint. So remember, sodium-potassium pump, pump sodium's out, potassium's in, active transport pump, moving things against the concentration gradient. More of the potassium ions leak out than sodium ions leak back in, so that therefore you have a positive charge outside. And also you've got lots of negatively charged ions inside in the cytoplasm. So the membrane is said to be polarized. Now, if we have small changes in the potential, as long as we're below the threshold level, we don't cause an action potential. So if we touch the Paxinian corpuscle very, very lightly, in fact, so lightly that we don't feel it because we don't generate an action potential, then we don't reach the threshold level. But as soon as you reach the threshold level, the voltage gated sodium channel opens and we start an action potential which rises to plus 40 millivolts. Now, this is the concept of all or nothing response, because remember, the transmission of an action potential, it's a digital signal. It's a 1 or a 0. The size of the stimulus doesn't change the amplitude of the action potential. It changes the frequency of the action potential. So it's digital. You either have one or not. Anyway, plowing on. So all or nothing law, the size of the stimulus is, size of the impulse, is independent of the size of the stimulus. If it's below the threshold level, you get no action potential. If it exceeds the threshold level, you initiate an action potential. And if it's a greater intensity, you don't get any greater action potentials, but you do get more frequent action potentials. I talked through all of this, I think. So depolarization is the sodium gates opening, and then the potassium gates reaching the top of the action potential at plus 40 millivolts. Then the potassium gates opening, the potassium's flooding out, and then repolarization, and then you're below the threshold level and it's hyperpolarized. And I've talked through all of that, so I won't go over that again. Okay, propagation of the action potential I talked about briefly when I was talking about with the whiteboard. So when you have an action potential, so at this point here, you're up at plus 40 millivolts because the sodiums have come flooding in. That influx of sodiums, then those sodiums move sideways down the axon to where there are less sodiums. And then on the outside becomes less positive because the positive charges move back along the axon, whereas the sodiums move along inside the axon. So at the next point along is going to become less positive on the outside and more positive on the inside, which will raise it above the threshold level, so it will cause another action potential at this point, so sodiums keep flooding in. And this is what propagates the action potential down the axon. And this is what's called the setting up of a local current, where the influx of sodiums by the action potential causes the depolarization of the next part of the axon, so the whole thing travels down the axon. Another picture of the local current here. So this is outside, this is inside. This is your voltage gated sodium channel opening. As a result, the sodiums come flooding in. We've got lots of sodiums here. We've got fewer sodiums at this point. The sodiums move along, which makes it more positive on the inside, which gets it over the threshold level. So then the voltage gated sodium gates open and cause an influx of sodiums, which propagates it down the axon. Okay, so node of Ranvier. Now, this does cause a little bit of confusion amongst students, so I'll just try and talk you through it. So when we've got a node of Ranvier, you've got axon, axon, and then you've got myelination. Now, your axons are very narrow indeed. They're only about 10 micrometers in diameter, yet you've got extremely high speeds of transmission. And the way you achieve those high speeds of transmission is that you miss out large chunks of the axon. And the bits where you miss it out are where the myelinated Schwann cells are. So the Schwann cells are wrapped around the axon and they contain myelin. So that's an electrical insulator. So it prevents an action potential from occurring at any of these points. Now the Schwann cells are very long cells. The Schwann cells are about 1 millimeter in length. And what's between the Schwann cells is a small unmyelinated gap, which is known as the node of Ranvier. Now, if we consider just as a resting potential, so at the node of Ranvier, that's going to be negative on the inside and positive on the outside because there'll be a resting potential. So you're going to be down here at minus 70 millivolts. Now, if there's an action potential at the previous node of Ranvier, then the sodiums will flood in, which will make the inside positive, and the outside will be negative. So we have here our action potential. Now, those positively charged sodiums will, in exactly the same way as the local circuit set up in an unmyelinated axon, those positively charged sodiums will be moved towards where it's negatively charged, and the positive charge on the outside will be attracted towards where there is a negative charge at the previous node of Ranvier. So this cloud of sodiums will move sort of towards this way, and this cloud will move sort of towards this way, which will mean that at this point, the next node of Ranvier, it will be more positive on the inside or less negative, and this point here, it will be slightly less positive. So what you do is that the potential difference at that point goes from being minus 70 millivolts to being above the threshold level. Now remember, the threshold level is very close to the resting potential, so you don't have to make the inside much more positive. You only have to make it by about 10 or 15 millivolts more positive, and then you open the voltage gated sodium channels, and then the sodiums come flowing in because you've reached the threshold level. Now, the effect of that is that you then miss out the entire millimeter of the myelinated Schwann cell, so that the depolarization that occurs at this node of Ranvier gets transmitted to this one and then bounces down the axon by saltatory conduction. Okay, I hope that made vague sense. So saltatory conduction, we get, and I've talked through it enough there, so I'll carry on. So factors increasing the speed of conduction down a nerve impulse. The biggest factor that's going to increase it in you is myelination. So we've got a node of Ranvier here, here, and here, and then these are the Schwann cells. And this here is the myelin wrapped around the membrane. And on the inside here, we've got the axon filled with axoplasm. So if you've got a myelinated axon, you can transmit action potentials at about 100 meters per second. In an unmyelinated neuron, it's only about 1 to 3 meters per second for the same diameter. So if you plotted diameter of the axon along the bottom and speed of transmission in meters per second, and what you get is this for an unmyelinated axon. Now, at about 1 millimeter in diameter, it's got a speed of transmission of about 100 meters per second. And that's what we see as speed of transmission in the squid axons, which are giant because they're not myelinated. But we have got axons that are only about 10 micrometers in diameter, yet we achieve speeds of transmission of around 100 meters per second. And that's because of the effect of myelination. Okay, so myelin is your lipid-rich layer, which makes the axon impermeable to ions. The ions can't diffuse in the tissue fluid from the tissue fluid into the neuron because they're blocked by the presence of the myelin sheath. So the action potential can't be generated in the myelinated region. So it has to jump its way from node of Ranvier to node of Ranvier, causing an increase in length of the local circuit. Diameter of the axon, this is something that tends to confuse students. And the point, this is a surface area to volume ratio. When you've got smaller axons, you would have proportionately more edge to the pipe, so edge to the axon, than if you have a larger diameter axon. It's a surface area to edge effect. So that would create more resistance. So if you have small tubes, you have greater resistance because you have proportionally more edge to the tube. Whereas if you have a big fat axon, you've got proportionately less edge, so you have less resistance to the flow of the ions. It's exactly the same when you're looking at the speed of flow of blood in capillaries and in veins. Remember, veins have got less resistance because you've got a very large diameter, therefore a big cross-sectional area in comparison to the edge of the vein. Whereas in a capillary, you've got a very narrow vessel, and that gives you a lot of resistance to flow. Temperature is the third thing that affects it. Increasing the temperature increases the production of ATP, and it also increases the rate of diffusion. So the sodium and the potassium ions can diffuse faster, so that nerve transmission is faster in endotherms. So produce more ATP and higher speeds of diffusion because of more kinetic energy. Okay, how are we doing? Everybody still with me? Yes, you are. Right, on to, let's look at a synapse. So now, with a synapse, you've got a change from an electrical signal to a chemical signal to another electrical signal. So a synapse is a connection between two neurons. We've got the arrival of an action potential at the presynaptic neuron. And then you've got some voltage-gated calcium channels. Now, these calcium channels open as a result of the arrival of the action potential, and that allows in calciums down the concentration gradient. That causes vesicles, and vesicles are spheres of cell membrane. Those vesicles are dragged to the presynaptic membrane, where they join with the presynaptic membrane in a process of exocytosis. That releases the neurotransmitter molecules into the synapse, and they then diffuse across the synapse, which is a very narrow gap, and bind to specific receptors on the postsynaptic membrane. Now they then open those receptors, and that allows through ions. And those ions may be sodium, which will then cause a rise in the potential on the postsynaptic neuron, or they could alternately, and this is something you may or may not have heard of, allow potassiums out, which would hyperpolarize the postsynaptic membrane, or alternately they might allow chlorides in, which would again hyperpolarize the postsynaptic membrane. Okay, so neurons connected together by synapses. So here we've got some connections coming into a neuron. And you can have inhibitory synapses, which make the potential more negative. So they hyperpolarize the postsynaptic membrane, therefore making a postsynaptic action potential less likely. Or you can have excitatory synapses, which make the postsynaptic membrane more positive, therefore make it more likely that you'll reach the threshold level and cause a postsynaptic action potential. So this is the start of information processing, is the relative frequencies of action potentials along inhibitory synapses along the neurons to inhibitory or excitatory synapses. So here's your classic synapse. We've got synaptic vesicles which contain neurotransmitter. There are lots of different neurotransmitters in your body. The one you need to know about is acetylcholine, but you do need to know there are others. The vesicles are full of neurotransmitter. We've got lots of mitochondria to produce ATP to power the exocytosis and to power the reconstruction of the ethanoic acid and choline back into acetylcholine after you've had an action potential. So arrival of the action potential opens the voltage gated calcium channels, in comes the calciums, causes the vesicles, which are spheres of cell membrane, to move to the presynaptic membrane. They become part of the presynaptic membrane, and that releases the neurotransmitter into the synapse, and then the neurotransmitter molecules diffuse across the synapse and bind to the postsynaptic receptors. So here we've got action potential coming down, vesicles joining, neurotransmitter getting released into the membrane, and then binding to a sodium receptor on the postsynaptic membrane. So this has a specific receptor for acetylcholine and causes the sodium ion channel to open, and as a result sodium ions go in, and that's because, remember, the inside is negatively charged because of the action of the sodium potassium pump, which has been pumping sodiums out and potassiums in again. So the sodiums flood it, come in. Now if sufficient neurotransmitter is released from the presynaptic neuron, and if it binds to sufficient postsynaptic receptors, then it may cause a rise above the threshold level, which may cause a depolarization of the postsynaptic neuron, because you've opened enough sodium channels to allow through enough sodiums to get it over the threshold level, which would then open the voltage gated sodium channels, which would then cause an action potential to spread down the postsynaptic membrane. So now we've got acetylcholine esterase. Now, it being called A's means it's an enzyme. So acetylcholine esterase hydrolyzes or digests acetylcholine into choline and ethanoic acid. Now that resets the junction, because those then diffuse back across the synaptic cleft, and then ATP is used to reform the choline and ethanoic acid back into acetylcholine, which is then restored in the vesicles. Right, here we go again. For a final time, arrival of the action potential allows in the calciums, causes the vesicles to move, releases the neurotransmitter, which diffuses across, binds to the postsynaptic neurotransmitter receptor, opens the ion channels, and allows sodiums to come in. The neurotransmitter acetylcholine then gets broken down by acetylcholine esterase, and the degraded neurotransmitter then diffuses back across and is reabsorbed into the presynaptic membrane, where it's then reassembled back into neurotransmitter. There are lots of different neurotransmitters. They can either be excitatory or inhibitory. If they're excitatory, they're going to make the postsynaptic cell more positive, so less negative, so make it more likely that you'll reach the threshold level. If they're inhibitory, they're going to make the postsynaptic cell more negative, so therefore making an action potential less probable in the postsynaptic neuron. A little bit more here about the receptor. So Here's your sodium receptor. We've got a binding site for acetylcholine. Acetylcholine binds and causes a conformational change to the sodium channel, so it allows through sodiums. And those sodiums move in by facilitated diffusion through the ion channel. The reason there's more they move in is because there's more outside, and that's been achieved through the sodium-potassium pump. No, exactly the same. So roles of synapses, now this comes up a lot in exams. So first of all, they make sure that the flow of the impulses is in one direction only. And that's because vesicles are only on the presynaptic membrane and the receptor molecules are only on the postsynaptic membrane. A very common question in exams: how does it ensure one-way transmission? It's the neurotransmitter is only released on the presynaptic membrane and the receptors are only on the postsynaptic membrane. Acclimatization, so after you've had repeated stimulation, it can run out of neurotransmitter. So if we have lots and lots of action potentials arriving at the presynaptic membrane, then you can run out of neurotransmitter. Integration, you can have an impulse traveling down one neuron and then that goes out to many postsynaptic neurons, and they can have convergence where lots come together and then one leaves. So that means that signals from lots of parts of the body can be integrated to create one postsynaptic action potential. Drugs, now this is a big topic. Now mostly exam boards look at drugs and the effect of drugs at synapses as a way of testing your understanding of synaptic transmission. So in the scaffolding to question, they tend to tell you enough that it's not a what question, but it's an explain using your knowledge of synaptic transmission to say how this drug could interfere with nervous transmission. So different ways that it can affect synapses: you could increase the number of impulses, you could increase the so you could cause the release of neurotransmitter from the vesicle without impulses. You could block the reuptake or block the receptors, so postsynaptic receptors. You could cause the production of more or less neurotransmitter, or you could prevent the vesicles from binding to the membrane and releasing the neurotransmitter by exocytosis. So excitatory drugs will stimulate the nervous system. Inhibitory drugs will reduce the transmission by creating fewer action potentials. One that does come up a lot is organophosphorus insecticides. So that inhibits acetylcholinesterase. And acetylcholinesterase is the one that breaks down acetylcholine, and therefore it gets reabsorbed back into the presynaptic neuron. Now, the problem with organophosphorus insecticides is that it is an inhibitor of acetylcholinesterase. Therefore, it prolongs the effects of acetylcholine. It remains in the synaptic cleft, and it remains bound to the postsynaptic neurons, postsynaptic receptors. So that means you have repeated firings of the neurons at the neuromuscular junction, and therefore there's permanent contraction of the muscle, and therefore you have paralysis and stopping of breathing. Okay, I'm just getting around to launching the next quiz. Okay, the next quiz is now running. It's on in the Socrative.com site. If you go there, Socrative.com, and put in A-level biology tutor, you should be entering the next quiz. So that's if you go to Socrative.com and put that in. I would stop this now. Actually, no, I'll go back to the beginning. So if anybody has any questions, please go ahead and ask them now. The quiz is running on socrative.com, A-level biology tutor. Where is the quiz? It's in socrative.com. I forgot to put the link in. I'm sorry about that. Sorry, Emily. Socrative.com. And if you put in A-level biology tutor, and it will take you straight to the quiz. And there's lots of you in there, all getting lots of good answers. That's great. Any more questions in the Q&A, people? So anything else you want me to talk about? Explain why the maximum frequency of the action potential is limited. I think that would be limited by the refractory period, which is the period under which the voltage-gated sodium channel won't respond. So after it's opened, because you're over the threshold level, it opens for about a millisecond, then it closes and it won't respond no matter what, which is known as the threshold value. Did that answer your question, Jay? Hopefully. Excellent. I'm pleased it has. Hope you're all in the quiz. Gosh, I can see lots of you in the quiz. That's really good. 6 questions in the quiz. Hope this has helped. Any more questions, people? How are we doing? Any more questions? Okay, well, next week it's protein structure and function, and the week after that it's osmosis and water potential. The week after that it's blood, hemoglobin and carbon dioxide transport. If you do want any other quiz, any other webinars, then drop me an email with a suggestion. I'm always pleased to hear. Where is the quiz? Everybody said thank you, and somebody had their hand up. I'm never quite sure what to do with someone when somebody puts a hand up. But if you've got a question, put it in through the Q&A box. So next week it's protein structure and function. The week after that it's osmosis. The week after that it's blood. And then after that I've got nothing planned. So if there's something you want me to do, then drop me an email or make a suggestion. Anybody got any more questions about action potentials and synapses, then please ask them now. So Jay, you said, I've got one more question. You were saying about the drug effect with acetylcholine being on the synapse. What effect does it cause? Well, the acetylcholine remains bound to the postsynaptic receptor. So the postsynaptic receptor remains open, which causes a constant depolarization of the postsynaptic neuron. So that would cause constant contraction and paralysis and death. No problem, Jay.